All right, we are here to talk about meiosis right now. We should have just watched the mitosis video that I put up a day or so ago, and now we're going to get into the process of reproducing to create gametes, or cells that are used for sexual reproduction. Some big stuff that comes along with meiosis is, first off, you need to know that there are two nuclear divisions. This is completely different than mitosis, which only have one division, so that's kind of one of the big distinctions between mitosis and meiosis. Chromosomes only get copied before that first nuclear division. So in that first interphase that occurs, that's the only time the chromosomes are copied, which is going to have an end result of a decreased number of chromosomes in the daughter cells compared to the original parent cell. This is by far one of the biggest things that we can say when we're talking about meiosis, is that we got 4 unique haploid cells are the result of meiosis. And that uniqueness is going to be a big thing when it comes to genetic diversity and everything sort of being different among the different gametes, the sperm or the eggs. So the 3 main goals when we're talking about meiosis are: 1, we got to go from a diploid parent cell to haploid daughter cells. After that, each haploid cell has to make sure they have exactly 23 chromosomes. And you think about it, your mom gave you 23 chromosomes and your dad gave you 23 chromosomes. And that's why we say that you are a diploid cell, because 1n plus 1n equals 2n. So each gamete has to be a 1n cell or a haploid cell. And the last goal of meiosis is that there's going to be genetic diversity created between all the gametes, because that can be very beneficial to the species as it goes through changing over time. Meiosis is broken down into meiosis 1 and meiosis 2, so we'll sort of talk about things that special features that go on during meiosis 1 and meiosis 2. The chromosomes only get copied before meiosis 1, and that happens during that S phase of interphase, after the cell's gotten its signal to move out of G1 and go on with the division process. And once it does that first division process, it's going to do the second process right over. So the only time the chromosomes are copied are before the first one, and that's kind of that's going to allow the cell to decrease from diploid to haploid. When we talk about some unique things that happen specifically in meiosis 1, you already know all the names of the stages. The names of the stages are similar to that of mitosis, in that you have prophase 1, metaphase 1, anaphase 1, telophase 1. Then you'll have in meiosis 2, prophase 2, anaphase 2, metaphase 2, anaphase 2, telophase 2. During meiosis 1, homologous chromosomes are going to line up together, and we'll talk about what homologous chromosomes are in a second, but they're going to line up together all duplicated. So if I have one set of chromosomes that was duplicated, that gives me 2 chromosomes that have the exact same DNA from my mom, and 2 chromosomes that give me the exact same DNA from my dad. And those 1, 2, 3, 4 chromosomes makes up your tetrad. In meiosis 1, the homologous pairs are going to separate, and during anaphase 1, and we'll talk about how that will create some genetic diversity in a second. Before meiosis 2 starts, the chromosomes do not get copied again, and we sort of mentioned that one a couple times, and again, that's to make sure that the resulting daughter cells are going to be haploid, and specifically, we're going to have 4 genetically unique haploid cells. That is by far the most important feature of meiosis, and one of the most important features of sexual reproduction, is that cells are unique, and that could provide offspring that are more fit for survival and reinforce good traits potentially in things that were fit to reproduce, because that is the purpose of biology, is to survive and to reproduce. So we talked about this idea a second ago, is what makes chromosomes homologous. Well, remember, you got 1 set of DNA from your mom, 1 set of DNA from your dad, 23 chromosomes in all. Homologous chromosomes are like chromosome number 1 from your mom and number 1 from your dad. They're both considered number 1 because they code for the same exact information. There's different genes, there's the same genes on the same chromosome from your mom and from your dad. So that's gonna make those ones homologous. You also, chromosome number 2 from mom and 2 from dad are homologous. 5 from mom, 5 from dad are homologous. Now if you looked at chromosome number 2 and number 6 from mom and dad, well, those would not be homologous. So when we're getting ready for prophase 1, the homologous pairs are going to bond together. So you have 4 different sister chromatids because they've been duplicated in the S phase, and they're going to form that tetrad. huge stuff that happens in prophase 1, and the biggest thing is going to be called crossing over. Prophase 1 is probably the longest phase of meiosis, and this is where the chromosomes are going to change, and this is where a lot of that genetic diversity is going to occur. The genetic diversity happens because those homologous chromosomes that are formed in tetrads are going to overlap on each other. And we can sort of see that going on right here, and the legs sort of overlap. And if you look, we see this red leg going down, and then it looks like the red leg actually switched with the blue leg. That process is called crossing over, where homologous chromosomes exchange genetic material. Where the chromosomes touch each other are called chiasmata. And this process could happen like 1 to 3 or 4 times between homologous pairs. Well, that means these things are going to separate eventually, and now this DNA has been changed. Granted, it's all coding for the same information, but your mom and your dad give you different forms of traits that are called alleles. So the alleles are going to get mixed up a little bit, and that's going to create genetic diversity, which is a huge, huge deal. Here's another picture of what's going on. This takes about a week in males for prophase 1 to occur, which, if we remember, mitosis takes like less than 24 hours sometimes. So this one phase takes a little bit of time, but it's going to ensure genetic diversity. Females, you guys actually go through meiosis before you're even born. You start creating your eggs in the womb as a fetus, and the eggs don't get released until you hit puberty, but they're already created and already ready to roll until you hit sexual maturity. So this meiosis process and crossing over are going to provide a lot of genetic diversity, which is extremely important. Genetic diversity leads to what we call independent assortment. And when we get to Mendel's ideas about how genetics are transferred from parent to offspring, independent assortment is another topic that we're going to bring up. But chromosomes are going to separate during meiosis independently of one another. Just because one of your mom's chromosomes goes to the left doesn't mean all of her chromosomes are going to go to the left. And so they might go left or right. There's a lot of different combinations that can be had based on that. If you do 2 to the 23rd power, it's a really big number. I think it's like 8 billion and change. So like every gamete is like 1 in 8 billion-ish. So that's a really big idea, and that's without the idea of crossing over, how many combinations there are. When you put the fact that all the alleles can get switched up through the process of crossing over in prophase 1, that can make basically an endless supply of unique gametes, which is a huge, huge deal when it comes to genetic diversity. So let's look at all the steps of meiosis in general. We'll start with meiosis 1. In meiosis 1, prophase 1 happens, well, the chromosomes are going to condense. The chromosomes condense up and they go through crossing over. Prophase 1 is the only time when chromosomes are going to cross over, so that's kind of a big important thing to know. Similar to mitosis, the centrosome is going to migrate to the poles of the cell, and that's going to transition us into metaphase 1. In metaphase 1, all the homologous chromosomes are lined up together, and doesn't matter how they're lined up. You might have the mom on the left or the mom on the right. It really doesn't matter. They're just lined up along the cell's equator with the centrosome at each pole of the cell. During anaphase 1, homologous chromosomes separate, and I like this picture because it's color-coded. You can see that these red chromosomes have a blue foot right here. That tells you that the chromosomes crossed over during prophase 1. This blue one has a red foot, meaning those two little portions just sort of switched with each other, and they're getting pulled to opposite ends. So I have one set of homologous chromosomes going one way, one set going another way. Now if you look here, I got red, red, blue. Down here I got blue, blue, red, and we'll just say that the red ones came from mom, blue ones came from dad. So you can see that they're independently assorting. Not all the blue ones are together, not all the red ones are together. After anaphase 1, then we go into telophase 1 and cytokinesis, where the cell actually splits and you get the new nucleus forming around the chromosomes. You can see that these guys have crossed over and the chromosomes are there's still 2 copies of them. Now the copies are a little bit different, however, because of that crossing over. In metaphase 2, the chromosomes line up right down the middle of each equator of each cell. In anaphase 2, now the centromere of those chromosomes that were duplicated is going to split, and one sister chromatid goes to each side of each cell. So now the sister chromatids are going to each side of each cell, and look at this. everything is just sort of jumbled up now. I have all different kind of arrangements of chromosomes going to each way. In telophase 2 and cytokinesis, the chromosomes uncoil, the new nuclear envelope forms around them, cytoplasm and organelles are going to divide, and that's going to lead to our 4 unique gametes. Now, that's kind of a lot of stuff going on. Do you think it happens right all the time? Not really. And when we talk about things that don't happen right, sometimes we can call those mutations. Now, don't assume a mutation is bad. Mutations just as often can be positive as they can be negative. We're going to talk about some negative ones, and we'll talk a little bit about some positive ones, because there are a couple that do help us out. But mutations can be one source of genetic diversity, which can lead to evolution over time. Non-disjunction occurs when the chromosomes don't separate correctly. So that could happen where sister chromatids don't, or homologous chromosomes don't separate. Maybe both homologous sets of homologous chromosomes move to one side, and that can result in something called an aneuploidy. Well, if both homologous chromosomes go to one side, one cell is going to have too much DNA, another cell is not going to have enough. And if we talk about DNA as being instructions for a house, you kind of want the exact amount of instructions there for you. You don't really want to have too much or too little. You want to have the exact right thing. When something is missing a chromosome, we call that a monosomy, because there's one area where the cell's missing a chromosome, and that's usually going to result in cell death. If that gamete were to fertilize with another gamete from a male or female, that might lead to a miscarriage, and about 10 to 30% of pregnancies have aneuploidies, which means about 10 to 30% of pregnancies never even go forward, and they result in miscarriages. Trisomy is when you have one extra chromosome. Now, that's not always, it's not good, but it doesn't always terminate the pregnancy. Sometimes, if they have trisomy at number 21 in particular, that's what we call Down syndrome. Trisomy 21 is another name for Down syndrome, which is a mental disorder that affects a person's brain development and some of their physical attributes. And that is a result of having one too many chromosomes at the 21st slot. Another thing that can happen is having polyploidy organisms. Polyploidy organisms are anything that's more than diploid. Plants, polyploidy occurs in plants quite often because a lot of plants will self-pollinate, but it can also happen in animals and may lead to speciation, which we'll talk about when we get to the whole evolution thing later on in the day. Basically, a lot of times with polyploidy, there happens to be an extra round of DNA replication, and that can create more chromosomes than are needed and can lead to more chromosomes. As long as the chromosome number is an even number, where you're 2n, 4n, or 6n, then usually that organism, if it finds another organism that has 4n chromosomes and you have 4n chromosomes, you could potentially reproduce successfully. If you have an odd number, that makes things a little bit more difficult. And when that happens, things become infertile. One popular way farmers use this idea of manipulating chromosome numbers when they make seedless plants. And on the right hand side of the screen, I have a picture of a watermelon, because seedless watermelon is a result of being triploid, where it has an extra chromosome in one spot, which leads to infertile, the watermelon being infertile. Well, the seeds are just used for reproduction for plants. I mean, when you think about that, the watermelon itself is just sort of a carrying case for those seeds, and you could plant those seeds and potentially grow a new watermelon. Well, if it's infertile, it's not going to have seeds, and that's where our seedless varieties come from. Translocation is another thing that can happen that gets a little wild. In translocation, non-homologous chromosomes are going to cross over. Hopefully remember that crossing over happens in prophase 1. Well, non-homologous chromosomes will be like in humans, number 9 from your mom and number 22 from your dad, or vice versa. If this happens, that can be something that gets passed down to offspring or affect you. If it happens during mitosis, well, mitosis happens to somatic cells, so that's going to cause problems with you, and that could be one way that leukemia would develop, is if there's translocation between chromosomes, numbers 9 and number 22. If that happened to one of your gametes, that's something that wouldn't affect you so much, but something you could pass on to your potential offspring. And that's obviously not a good thing. We don't want to pass down potential disorders to the offspring. That's not something that we look forward to try to help the species out. One way we can look at chromosome numbers and see if there is any issues with that is doing a karyotype. And basically a karyotype is a way where chromosomes have a picture taken of them. And based on that picture, a computer program will separate out all the chromosomes, sort of pair them up so you can see the chromosomes that you're supposed to have, and you can tell if all the chromosomes are there. And this is a really quick and easy way to figure out if someone's going to have Down syndrome or not before they're born. You can do a karyotype on the fetal, on the fetus's DNA. When we look at a karyotype, we sort of see them out right here. We got chromosomes 1, 2, 3, 4, 5 across the top, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23. Now the karyotype separates these chromosomes based on size and shape. There's one exception, and this is, if you look at chromosome number 23, this is what we call our sex chromosome, because that determines whether you're a male or a female. Females are going to have 2 X chromosomes. Males will have an X and a Y. The Y chromosome is going to look a little bit different than the X chromosome, which if we look at this karyotype, this picture is showing us a male because I see one chromosome is a little bit longer than the other one, so I see 2 different ones. Unfortunately, gentlemen, our Y chromosome doesn't do too much other than tell us how to make sperm, so not really too helpful in anything else, but hey, it does a good job at that. If we look at this karyotype, we should see a little bit of an issue. We can see all the chromosomes are numbered 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23. This is going to be a female since they're both the same size. They're both X chromosomes. But if we look over here at 21, you'll notice 1, 2, 3 dots right there. That should tell you that there's a trisomy at 21, which if you remember from a couple slides ago, a trisomy at number 21 would lead to Down syndrome in this patient. So we've kind of gone through a lot of stuff with meiosis. We went through the phases really quick. We went through a couple key differences in between meiosis and mitosis and sort of what's going on. Hopefully you enjoyed this, and I will talk to you soon.